Second, they're going to determine if Fuse Binding Protein1 affects hepatitis C replication cell division, so they want to learn more about its role. And then lastly, they're going to determine if it interacts with p53, a known tumor suppressor. So the first thing they want to answer is the Fuse Binding Protein1 present in all liver tumors or only those with a history of hepatitis C? So to do this, they use immunostaining. What this is that they used a bunch of different stains on 3 different types of liver cancers. One, on the top 2, are caused by hepatitis C. The second one is caused by alcohol, and the third one is caused by liver disease. So there's multiple ways someone can get liver cancer. Hepatitis C is just one of them. And then they use 3 different types of stains. On the first column, they use the Fuse Binding Protein1 antibody, which detects whether or not there's the Fuse Binding Protein1 present. The second one they use is the isotype IPG, which just checks to see if there's any background stain. And then the third one is H&E, which checks to see if there's cancer present. So you can see in the first column that the Fuse Binding Protein1 was only present in those caused by hepatitis C, which are the first 2 rows, and not in those caused by alcohol or liver disease. And then in the isotype IPG column, you can see that there's not much there, which is good. We don't want background staining interfering with our data. And then the last one, those purple dots indicate that there's cancer in all 4, which is what you'd expect because they're all liver cancer. So what we can conclude is that the Fuse Binding Protein1 is only present in liver cancers with a history of hepatitis C. Any questions? So now they want to know more about its role. Like, does it affect hepatitis C replication? So to do this, they used a gel which showed the amount of hepatitis C replication. In the first column, the cured, there isn't any replication. In the control, and then in the knockdown, there's decreased hepatitis C replication. And then when they overexpressed Fuse Binding Protein1, there was a lot of hepatitis C replication, which shows that the Fuse Binding Protein1 is likely, it causes more hepatitis C replication and it has some interaction there. And then on the right, you can see a quantified version of the same data. Anyone have any questions? So now that they know that it does cause more hepatitis C replication, they want to see if it has any, if it causes tumor growth. So to do this, they used a wound healing assay. Basically, like seeing how a wound fills in is an easy way to visually see cell division. The top column is a control, so that's just a normal wound being filled in. And then the second one is the Fuse Binding Protein1 knockdown. And they simulated a wound by making scratches in the cells and then watching the cells grow back. And so we can see in the control that the wound filled in a lot faster. After 32 hours, it's almost completely filled in. Whereas the Fuse Binding Protein1 knockdown, it's still, there's a large wound in the middle. So that shows that the Fuse Binding Protein1 causes cell division. And then on the right is just the same thing, except it's like numbers rather than just visually, so that there's both like qualitative and quantitative data. Anyone have any questions? So now that they know that the Fuse Binding Protein1 causes tumor growth, they want to see if it does this through p53, the known tumor suppressor gene. So to do this, they used Oncomine data analysis. Basically, it's just a cancer database, and they were looking to see if it had any similarities. So they checked for 19 normal human livers, 59 cirrhotic livers, which is just liver scarring, and then 38 liver tumors. And they found that among these, they showed a 4-fold increase in Fuse Binding Protein1 expression and a 2.5-fold decrease in p53 expression. So you can see in the first graph, it's the Fuse Binding Protein1 expression. We can see that in cirrhosis and HCC, which are the 2 caused by hepatitis C, there's a 4-fold increase in Fuse Binding Protein1 expression. And then in the right graph, which is p53 expression, there's a 2.5-fold decrease. And they just wanted to see if there was any difference between cirrhosis and hepatocellular carcinoma, like the liver scarring, liver cancer, and they didn't find a difference. So that was just one other thing they found here. Does anyone have any questions? So now that we know that people, yeah.
Would you go to the previous slide?
Of course.
And this is just, I'm just thinking this out loud. When we start looking at the first graph that has the FBP1 messenger RNA expression, you're going to have low amounts in the normal cells, so there are higher amounts of p53 messenger RNA being expressed. Is that indicating that it's somehow protecting and it's a healthy cell? Versus when you see HCC, it's very high in FBP1 expression versus very low p53 expression?
Yeah, the HCC is like liver cancer, so it isn't working to protect your cell, so that's why it's decrease p53 expression, which would lead to tumor growth. And basically what this data shows is that the Fuse Binding Protein1 likely does a lot of things through p53 and it inhibits p53 to cause tumor growth. And the next thing they're going to look at is if it affects hepatitis C replication. So they want to see if Fuse Binding Protein1 causes more hepatitis C replication through p53. So to do this, they used a p53 knockdown. So we can see in the first section, they used a control. In the gel, you can see that the p53 is present. In the second section, there's a p53 knockdown, which the gel shows that there isn't any p53 expression, or very little. And then when they restored the p53 in the knockdown, it went back to normal. And we can see in the graph that when they knocked down p53, it caused a lot more hepatitis C replication, which shows that the Fuse Binding Protein1 likely causes more hepatitis C replication through inhibiting p53. And also, you can see in the column there's a p21 there too. That was another part of the paper, but I didn't think it was that important, so I didn't address it in this presentation, but that was just another correlation they found. Does anyone have any questions? So a recap of all the things that they found. First, they found that the Fuse Binding Protein1 is only present in liver cancers with a hepatitis C replication, with a hepatitis C background, which means that it's only in livers caused by hepatitis C. Next, they found that the Fuse Binding Protein1 causes increased hepatitis C replication and cell division, and it does this through inhibiting p53, a known tumor suppressor. So the next steps in this lab are to find the exact sequence of p53 that the Fuse Binding Protein binds to. And Dr. Pandey, the professor I worked under, divided the p53 gene into different pieces, and he's going to test each piece to see what binds to the Fuse Binding Protein and narrow down to the exact nucleotides. So my part this summer was I isolated a piece of the p53 gene, and then after that I left, the PhD students are going to see if it binds to the Fuse Binding Protein1.
Do you know if these cells were human cells? Like the ones
Yeah, they were. So the bigger picture is that if they can find an easier way to cure hepatitis C, it could make a big difference for a lot of developing countries in South Africa, Asia, and Africa, where there's high prevalence of hepatitis C due to unsanitary needles and blood transfusions. So it could save a lot of lives, and especially for people who don't know that they have hepatitis C and let it prolong into cancer, this could be a helpful research for them. Thank you. Does anyone have any questions?
Thank you.